Okay, in this video tutorial we're going to finish off genetic control and talk quickly about evidence for the semi-conservative nature of DNA replication. So before we do that, let's just quickly talk about what does that mean, or just quickly recap DNA replication. So we spoke about how the semi-conservative nature of DNA replication is the idea that the original parent strand, so you can see that on the top and the bottom here, is split in half, and that free nucleotides are added to match up the bases to synthesize a new strand. So one thing I didn't mention was these free nucleotides often have 3 phosphates attached to them. So a nucleotide triphosphate, and that may already maybe ring a bell in your head. So the major source of cellular energy in a cell is ATP, which actually stands for adenosine triphosphate. So that triphosphate is often a sign of something that's high energy. And so 2 of the phosphates are released as this bonds, and as you know, or as you can even see here, they've tried to show in the background this alternating, the yellow circle's phosphate, so alternating, sugar, phosphate, sugar, and the bases are the bit in the middle. So here you can see sugar, they've shown as the bits in the backbone, with the phosphates alternating in between the little yellow bits, and the bases are the coloured bits sticking into the middle. So we use nucleotide triphosphates, nucleotides with 3 phosphates on them, are used because that's the source of energy that is for the reaction. 2 of those phosphates are released, and the remaining phosphate is covalently bonded onto the OH, the 3' OH end of the exposed strand, that exposed OH on the 3' end of that strand. So it's always onto this OH end where a nucleotide is added. And so remember, that's essentially why. So nucleotides can only be added to a 3' hydroxyl end, and that's why DNA polymerase can only read DNA in a 3 to 5' direction, and synthesize DNA in a 5 to 3' direction, so complementary to that. So on the lagging strand, because it can only go in that direction, it has to read 3 to 5', it has to copy the DNA in short bursts as the DNA continues to be opened up by helicase. So if you're not sure what I'm talking about, you probably didn't watch the last video in this playlist, so go have a look at that. Anyway, let's talk a little bit more about semi-conservative DNA replication. So these were the 3 suggested models at the time. So semi-conservative, the idea was the strand is split, and we know this is today that this is the true mechanism, that the DNA strand was split into 2, and that a new strand was synthesized that was complementary. So you could imagine breaking the hydrogen bonds, you've got 2 red strands, and the pink one here has kind of been synthesized just by matching up the bases. A fully conservative replication would mean that essentially the 2 strands are kept together or intact. So here you end up with DNA that is half old and half new, whereas here, conservative, you end up with a strand that is entirely made up out of the original DNA and a copy is made separately. Now, scientists suspected it would be semi-conservative because it doesn't seem to make much sense to do this. This would be much more prone to mistakes. And it seems like the double-stranded nature already suggests that the semi-conservative method would make a lot of sense. So you could simply split the strands and then match up the bases to make the complementary strand. Dispersive was the idea that possibly even the DNA might be split into kind of chunks, and that some parts would be original DNA and some parts would be the newly synthesized DNA. So scientists suspected it would be semi-conservative, but obviously, someone had to find evidence for that. So that's essentially where these 2 guys come in. So this is these are the two experimenters go by the last name of Meselson and Stahl. Probably they're 2 American scientists, but the names probably have European roots, so possibly Meselson and Stahl. But 2 American scientists who basically were able to prove or find evidence that it is semi-conservative. And their experiment was actually fairly simple. It basically went like this. They fed some bacteria nutrients or foods that only contained a radioactive isotope, a slightly heavier isotope of nitrogen, nitrogen 15 or N15. So this isotope of nitrogen is slightly heavier. It's not. I don't think it's dangerously radioactive, but it's just slightly heavier. Has a slightly greater molecular mass because it's got that extra neutron than the nitrogen 14, the regular nitrogen 14. So they'd feed this to bacteria and in a culture, and then they'd put them in a tube which has a density gradient. So it has a fluid in here. So often I know, for instance, you can use a sucrose gradient where there's more sugar dissolved, so it becomes more thick and heavier, like a syrup down the bottom, and slightly less dense at the top. Now this one wasn't sucrose, I can't quite remember off the top of my head what it was, but they set up a density gradient, a fluid where it gets denser as you go further down the tube, and placed some of the bacteria in it, and then spun it in a centrifuge. And so the density or the molecular weight of the bacteria or their DNA rather would determine where that DNA would be found. So they started off. They fed bacteria with nutrients that only contained nitrogen 15. And after many generations, so they let them grow for lots of generations, the E. coli essentially had all of the nitrogen in their DNA. Now remember, DNA has nitrogenous bases in it. So all of the nitrogen in those bases was nitrogen 15. So almost 100%, or so close to it that we can basically say 100% of the DNA contained nitrogen 15. And so what happened is, if they spun this down a centrifuge, nitrogen 15 is slightly heavier than its regular nitrogen 15. So they got a band. All the DNA extracted from those bacteria was found down here. So that was after all of the bacteria had been grown with this nitrogen 15. Then what they did is they introduced, they took away that nutrient source, and they introduced a nutrient source that only contained the regular nitrogen 14, the regular version of nitrogen, which is slightly lighter. And in the first generation here, and that's probably one of the really key parts of the experiment, in this first generation here, all of the DNA was slightly lighter. And that's because every strand that contained, now I'm going to do every strand that contained the original nitrogen 15. So you can imagine the original DNA, sorry, let me clear this, in this population here was all really heavy, was slightly heavier or more dense, because both strands were contained the nitrogen 15, the heavier isotope of nitrogen. Whereas once the nitrogen 15 was taken away and the bacteria only had the nitrogen 14 available, in the next generation, well, each of these strands of DNA were split in half. And a new copy, so I know that's pretty faint, but the yellow there trying to show was synthesized using the nitrogen 14. So you can see that you'll end up with two copies in which essentially that are half original DNA, the heavy nitrogen 15 DNA, and half new DNA, the lighter nitrogen 14 DNA. So all of the strands of DNA were slightly lighter, and that was already evidence that it was a semi-conservative process. But if we look at the next generation, so still being fed purely now with the nitrogen 14, what would happen if these divided again? Well, what happens now is if we just take one of these strands to kind of show you, what happens now is that strand splits, and we get a red strand and a yellow strand. And so in this case now, remember we've only got the lighter nitrogen 14, so on the red strand we get a yellow, the lighter nitrogen-14 strand, and on this one, the one that's already lighter, we get a lighter version as well. So now we see two bands appear because we've got two sets of DNA in there. We've got some that here, so this is DNA that's entirely made containing the heavier nitrogen-14. Here all the strands are half old, half heavy nitrogen-15 and half newer nitrogen-14, so they're an intermediate weight. And here we start to get two bands. These are half of the DNA strands. They're the half of the DNA strands that you have here. They're still the ones that are a medium weight. So they're roughly in the same position as that previous band. But half of the strands now are contain entirely the nitrogen-14, the lighter version. And so half of the DNA is now at a slightly higher position, indicating that it's slightly higher or slightly less dense. And so after another generation, if we go through another generation, again, you've got half of these strands half of the daughter strands or the resulting strands from these will be completely yellow, and all of the ones from the ones that were already completely yellow, completely containing nitrogen-14. And so I'll draw it. I'll do this one. So this one here will split. Into a red and a yellow strand. The yellow strand here will just split into two yellow strands. And then basically now we synthesize new copies, and because we've only got the lighter nitrogen-14 available, they're all going to be yellow strands. So draw it like that. So now you can see that we've got four strands here, four copies now, and you can see that one of them is half old, half the original heavy, and half new nitrogen-14, and the other three, they're half old from this previous generation, but they're entirely nitrogen-14, the lighter. So this band here gets thinner because we now only have a quarter of the strands that are that slightly or that medium weight, whereas 3/4 of them now are the lighter nitrogen-14 version. And if you keep doing this, essentially you start to dilute out those heavy strands, and the band gets thicker and thicker here until eventually this band here is no longer visible. This one down here. So this is essentially evidence of the semi-conservative nature of DNA replication. It's kind of cool that without being able to see the DNA, we still were able to find evidence of what was going on there. So here, E. coli or bacteria fed a nutrient source that contains only nitrogen-15, a heavier isotope of nitrogen, so all their DNA became heavy. And then once they were fed the regular isotope again, after one generation, half of those strands were split, and so all of the DNA now was half heavy N15 based, and the other part contained the lighter N14. So these were the hybrid strands. And then after that, some of those hybrid strands, we start to get more and more strands that are entirely that yellow, that N14. So I hope that kind of makes sense. That explains these bands. And if you're doing the Cambridge exams, I believe you can be asked to explain what happens in terms of these bands. So there are only ever three positions: the starting position, where it's purely the heavy DNA. Here you've got the hybrid DNA, and here you've got the third position as the lighter DNA, or the N14 DNA. So cool, I hope you found that helpful, and that's it for the genetic control topic. I do sort of still wonder why DNA replication is in the genetic control topic, but we'll get on with the nuclear division, which ties in nicely with the DNA replication. So I guess we'll look at in the next videos why DNA is replicated, and the different types of cell divisions that occur in which DNA is replicated.